A very good morning to all of you students. Today we are going to discuss about cellular events of acute inflammation. I am Dr. Bharathi, Assistant Professor, Department of Pathology, SLIMS. So having discussed about vascular events in our last class, today we are going to discuss about cellular events of acute inflammation. So what are these cellular events? Cellular events are nothing but the neutrophils and macrophages which are capable of phagocytosis, or other WBCs or leukocytes which are capable of phagocytosis traverse these endothelial cells and go to the site of injury where there is microorganisms or other toxic injurious agent, and phagocytose them and eliminate them from our body. So the cellular events can be classified into following heading. That is increased influx of neutrophils in the vessels. That is the first step. The second step is margination. Margination is nothing but the neutrophils and the macrophages which accumulate in the margin of the blood vessels. The second step is rolling. These leukocytes which tumble over the endothelial cells. Rolling can also be called as weak adhesion, because when they tumble from one endothelial cell to another endothelial cells, there is a weak adhesion that is happening. The next is adhesion, which is otherwise called as firm adhesion. There is firm adhesion of the endothelial cells to the leukocytes. Leukocytes are adhering very firmly with the endothelial cells. The next is transmigration or diapedesis. The word itself tells that the WBCs or leukocytes which breaks the basement membrane and it comes out and it transmigrates to the site of injury. And finally, how this transmigration occurs from the blood vessels, it comes out of the blood vessels. After that, it has to move to the site of injury. This movement occurs with help of chemotaxis. Chemotaxis is nothing but there are certain chemical mediators which are secreted which attracts the WBCs to the site of injury. And finally, there is phagocytosis. Okay, phagocytosis is nothing but eating up of these WBCs. By eating up of these bacteria and microorganisms by the leukocytes is called as phagocytosis. So, this process delivers leukocytes capable of phagocytosis, that is, the leukocytes which are capable of phagocytosis or neutrophils and macrophages to the site of injury. So, these events can be divided into two types. One is leukocyte recruitment and leukocyte activation. So, the cellular events can be classified into leukocyte recruitment and phagocytosis. Normally, the leukocytes move rapidly in the blood along with the WBCs. The corner of the blood vessels is usually accumulated by the plasma. So, this is called as normal lamellar axial flow. That means the RBCs and WBCs will flow in the center, and periphery there will be plasma. So, the leukocyte extravasation is the process of migration of leukocytes from the lumen of the vessel to the site of injury in the extravascular tissues. So the steps in leukocyte recruitment or extravasation can be classified into four types, as I told you in the beginning of my class, that is margination, rolling, adhesion of leukocyte to the endothelium, transmigration or diapedesis. Point to be noted, transmigration is otherwise called as diapedesis. It can be asked in your MCQ questions also. So in normally flowing blood in the venules, the erythrocytes and the leukocytes accumulate a central axial column, which is called as normal lamellar blood flow. What happens in case of inflammation? What have you studied in vascular changes of acute inflammation? What happens? There is vasodilation. So whenever there is vasodilation, there is leakage of plasma to the extravascular space. So what happens? The WBCs and RBCs in the center will accumulate, will come to the margin of the blood vessels. Usually the WBCs are larger than RBCs, so they come to the margin of the blood vessels. So that process is called as margination. In inflammation, there is stasis of blood. So the hemodynamic conditions and more white cells assume a peripheral position along the endothelial surface, which is called as margination. So the rows of leukocytes tumble slowly along the endothelium and they adhere transiently. That is called as rolling. The rolling is otherwise called as, we can call it as weak adhesion. They tumble from one endothelial cell to the another endothelial cell by weak adhesion, which is otherwise called as rolling. Okay, they rest at some point and adhere firmly. Okay, firmly to the endothelial cells. That process is called as adhesion. This resembles pebbles over which a stream runs without disturbing them. So this process is called as adhesion. After firm adhesion, the leukocytes insert pseudopods into the junction between the endothelial cells. How does this leukocytes inside the blood vessels comes out of the blood vessels? By inserting pseudopods into the junction between the endothelial cell. That is, they break the basement membrane of the endothelial cell. They squeeze through the endothelial junction, and they lie between the endothelial cell, which lies between the endothelial cells and the basement membrane. So they squeeze this junction and they come out of the blood vessel. That is, they come out to the extravascular space. So now they traverse the basement membrane and they escape into the extravascular space. All the WBCs like neutrophils, monocytes, lymphocytes, eosinophils and basophils use the same pathway to migrate from the blood into the tissues. Now let us see each and everything in detail, each and every point, that is how the rolling occurs, how the margination, how the rolling, how adhesion, transmigration, everything occurs. Okay. So how the leukocyte gets adhered to the endothelial cells? There should be some mechanism, right? So there should be some molecule on the surface of WBCs which attract the molecule which is present on the surface of the endothelial cell. So these molecules which helps in attracting the WBCs to the endothelial cells are called as selectin. So these molecules helps in weak adhesion or rolling. The next molecule which helps in firm adhesion is called as integrin. That helps in firm adhesion of leukocytes to the endothelial cells. And there are certain molecules which helps in transmigration. And after transmigration, what happens? The WBCs are attracted to the site of microorganisms or to the site of toxic injurious agent. So what is that? They are called as cytokines or chemokines. Chemokines are nothing but they are the chemical mediators which attract the WBCs to the site of organism or to the site of injury. Okay, so leukocyte adhesion and transmigration occurs by binding of complementary adhesion molecule on the leukocytes and the endothelial surfaces. And there are also certain chemical mediators which are called as chemoattractants or cytokines. By modulating the surface expression or ability of such adhesion molecules. One more point to be noted here is that these molecules, that is the surface molecules, will not be expressed on the normal WBCs or endothelial cells all the time. They will be expressed only at the time of inflammation. They will be expressed only at the time of inflammation, or they will be expressed very weakly in the leukocytes. At the time of inflammation, there will be surface expression of these adhesion molecules. Okay. So, as I told you earlier, this is the leukocyte. The leukocyte has certain adhesion molecules which are called as selectants. So, I'll talk about selectants in detail. There are three selectants. One is P-selectin, E-selectin and L-selectin. P stands for platelets and endothelial cells. So, these P-selectants will be expressed on the surface of platelets and endothelial cells. L-selectin will be expressed on the surface of leukocyte. And E-selectin will be expressed on the surface of the endothelial cells. So P-selectin and E-selectin will be expressed on the surface of the endothelial cell, and L-selectin will be expressed on the surface of the leukocyte. Clear? Now, if there is expression of P-selectin and E-selectin on the endothelial cells, the corresponding ligand should be expressed on the surface of leukocytes. What is the ligand which is expressed on the surface of leukocytes? That is Cellular X modified glycoprotein. This is the glycoprotein which is expressed on the surface of leukocytes, which binds to P-selectin and E-selectin. Can you note down from this picture that is Cellular X modified glycoprotein is binding to P-selectin and E-selectin. Similarly, there is presence of L-selectin on the leukocytes which bind to certain molecules on endothelial cells, which I will be dealing with in detail. Okay, this is all about selectin. That is, selectin is the molecule which helps in rolling. Students, remember, selectin is the molecule which helps in rolling. Next, I told you in the beginning of my class, integrants or the molecules which helps in firm adhesion. How does these integrant molecules get expressed? These integrant molecules are activated by certain chemical mediators which are called as chemokines. These chemokines express the integrants which are like expressed weakly on the surface of WBCs. So these integrants are expressed weakly on the surface of WBCs. Whenever there is inflammation, there is chemokine. So these chemokines activate these integrants and they are strongly expressed on the surface of the leukocyte, which helps in firm adhesion. Note one point, the selectants are present on the surface of endothelial cells as well as the leukocytes, whereas integrant molecules are present on the surface of WBCs. The corresponding integrant molecules, so whenever there is a molecule, there should be ligand for adhesion. So the integrant molecules, there are two types, beta-1 integrant and beta-2 integrant. So the corresponding molecule will be located on the surface of endothelial cell. So which helps in stable adhesion. Okay. So for rolling, selectants are essential. For adhesion, integrants are essential. And for migration through the endothelium, we have PCAM1, that is CD31, platelet endothelial cell adhesion molecule, which is otherwise called as CD31, which helps in breaking the endothelial cell, inter-endothelial cell junction and the basement membrane, and it comes out of the endothelial cell and it reaches the extravascular space. When this WBC reaches the extravascular space, some of the chemical mediators which are secreted attracts these leukocytes to the site of microorganism. So this process is called as what? Chemokine or transmigration. The transmigration of these leukocytes to the site of injury occurs with the help of certain chemokines. After transmigration, that is the WBCs has reached to the surface of the microorganism. What happens next? There is phagocytosis of the microorganism. Phagocytosis of the microorganism. So phagocytosis is itself a big chapter. I will talk about this in my subsequent slides. So as I told you earlier, margination, when the blood flow slows down, the leukocytes move towards the peripheral column and accumulate along the endothelial surface of the blood vessels is called as margination. Next process is rolling. The marginated leukocytes attach weakly to the endothelium. They detach and bind again with a mild jumping movement, which is called as rolling. And the molecules involved in the rolling is selectin family of adhesive molecules and its complementary ligands. So I have told you earlier, there are three selectins, L-selectin, E-selectin and P-selectin. L stands for leukocytes, so they are expressed on the surface of the leukocytes. E stands for endothelium, so they are expressed on the surface of the endothelium. And P stands for platelets, so they are expressed on the surface of endothelium as well as the platelets. So the corresponding ligand is Lewis X ligand, which is expressed on the surface of the leukocyte. The corresponding ligand for E-selectin and P-selectin is expressed on the surface of the endothelium. Similar way, the ligand for L-selectin, which is expressed on the surface of the endothelium, is glycam-1, matcam-1. Glycam-1 and matcam-1. You remember these two. That is it. Okay. So, the similar way, the integrals, this is selectin and ligands, this is given in your Robbins. You have to mark this table up for your exams. That is selectin and the selectin ligands, integrant and the integrant ligands. Okay. So, the integrant ligands, I told you, integrants are expressed on the surface of what? WBCs. So, the corresponding ligand should be expressed on the surface of the endothelial cells. So, BLA4 integrant is otherwise called as beta-1 integrant, and the corresponding molecule which is expressed on the WBCs is BCAM1. And CD11 bar CD18 integrant is otherwise called as beta-2 integrant, and the corresponding ligand which is expressed on the WBC is ICAM1. Okay, then for transmigration, there is CD31 which is expressed on the endothelial cell, that is PCAM. Okay, that helps in transmigration of leukocytes through the endothelial cells. So as I told you earlier, all these integrants and selectants will be underexpressed. Only during inflammation, these will come out. So the regulation of endothelium and leukocyte adhesion molecule, where this P-selectin is seen, I've told you it is seen on the surface of the endothelium, but actually not. This P-selectin is seen inside the endothelium. There are certain molecules which are called as Weibel palade bodies. Certain molecules which are called as Weibel palade bodies. This p-selectin is present inside these Weibel palade bodies. Only during inflammation, I told you certain chemical mediators are secreted. The chemical mediators like histamine and thrombin, which helps or which makes these p-selectin, which is present intracellularly, to come out and express on the surface of the endothelial cells. Surface of the endothelial cell. That is how the p-selectin is expressed on the surface of the endothelial cell. Clear? So selectives are either not present or expressed at low levels in unactivated endothelial cells. The selectin and its complementary ligands are responsible for rolling. So the chemical mediators which are involved in expression of these selectins which are present intracellularly to the surface are tumor necrosis factor, interleukin-1 and certain chemokines which are called as chemoattractant cytokines. The next is adhesion. What is adhesion? This is just I am telling again about all these things which I've talked in the beginning of my case. Adhesion is nothing but the endothelium gets activated and leukocytes bind more firmly. Adhesion is nothing but firm adhesion. Rolling is just weak adhesion and it is a transient adhesion. Clear? The molecules which are involved are integrants and the corresponding ligands. So integrants are responsible for firm adhesion of leukocytes within the endothelial cells. Okay? So you have to mark up this. Next, coming to transmigration or diaperesis. The leukocytes migrate through the vessel wall by squeezing through the intercellular junctions between the endothelial cells, and the molecules involved is CD31 or PCAM, platelet endothelial cell adhesion molecule. So the migration, it migrates across the basement membrane with the help of an enzyme called as collagenase. Basement membrane is made up of type 4 collagen. So the leukocyte has to digest this type 4 collagen in order to come out of the blood vessel. So the collagenases are the enzymes which helps in breaking of the basement membrane and coming out. So what happens outside the vessel wall? Outside the vessel wall, there is chemotaxis. The WBCs has reached outside the vessel wall, so it has to move towards the microorganisms. So what happens? There are certain chemical mediators which attracts these WBCs to the site of bacteria. So that process is called as chemotaxis. Chemotaxis is defined as the process of migration of leukocytes toward the inflammatory stimulus in the direction of the gradient of locally produced chemoattractants. Chemotaxis is the unidirectional movement of leukocytes towards the injurious agent. It is a unidirectional movement. It moves only in one direction. That is, the WBCs moves only towards the microorganisms. It won't wander anywhere else. Okay, so it is a unidirectional chemical gradient. Because of chemical gradient, it is moving to the site of inflammation, or to the site of injurious agent. So what are all these chemoattractants? What are all the substances which attract the WBCs to the bacteria? It can be divided into exogenous and endogenous. The exogenous substances are the bacterial products. There are certain products which are formed on the surface, which are in the surface of the bacteria, like N-formyl methionine termin amino acids. So these products also attracts the WBCs towards the site of injury. And endogenous products such as leukotriene B4, interleukin 8, C5a and C5-3. So these also helps in attracting the bacteria to the, attracting the WBCs towards the bacteria. So how this happens? The chemotactic molecules which I told you earlier binds to specific cell surface receptor. Binding results in G protein mediated signal transduction event. So there is increased cytosolic calcium. So this cytosolic calcium helps in assembly of cytoskeletal contractile elements which are necessary for movement. So thus the leukocyte move by extending philopodia or pseudopods. I told you, it extends pseudopods and it moves in front because of increased cytosolic calcium, and it helps in assembly of cytoskeletal contractile elements which are necessary for movement. Okay. Now I'm just summing up. For rolling, the molecules which are essential is selectin. For adhesion, the molecule which is essential is integrant. For transmigration or diaperesis, the molecule which is essential is PCAM or CD31. And for chemotaxis, the molecules essential is, there are so many things, endogenous and exogenous. Exogenous is the bacterial product and the endogenous is leukotrienes, C5a, and interleukins. Okay, you have to mark it up. Next, the leukocytes should get activated. The leukocytes has reached to the site of bacteria. So for then the leukocytes should get activated and it has to recognize the bacteria. Okay, now the leukocytes have reached to the site where the bacterial organism or the microorganism is. After reaching there, it has to identify that this is the microorganisms and it has to get activated. So that process is called as leukocyte activation. Leukocyte activation is nothing but recognition of microbes or dead cells by the leukocyte receptors, which initiate several responses in the leukocyte, together known as leukocyte activation. The most important functional responses of leukocyte activation is phagocytosis and intracellular killing. So recognition of microbes or necrotic cells or foreign substances which require certain receptors on the surface of WBCs or the microorganisms. These receptors are called as pattern recognition receptor. This would have dealt in the beginning of the class. Toll-like receptor and inflammasome or the pattern recognition receptor. So the microorganisms are identified by the WBCs by certain receptors called as pattern recognition receptors. I told you these are toll-like receptors and inflammasome. After recognition, it has to phagocytosis. Phagocytose these microorganisms. What is phagocytose? In a simpler term, phagocytosis is nothing but eating up of the microorganisms and killing it. Okay. So now, the phagocytosis and release of enzymes by neutrophils and macrophages has the following step. What are those following steps? Initially, it has to recognize and attach. It has to recognize and attach the microorganisms. Now the WBCs has reached to the site of the bacteria. After reaching there, it has to recognize the bacteria and it has to attach to the surface of the WBCs. After attaching, it has to engulf the bacteria. After engulfing, what happens? It has to kill or it has to degrade the ingested material. Okay. So now, as of now, we have learned the 4 steps, 4 cellular events. What are the 4 cellular events we have learned? This is just brushing. First is margination. Second one is rolling. Third one is adhesion. Fourth is transmigration or diapedesis. Fifth, phagocytosis. So, margination, as you all know, because of vasodilation and because of leakage of plasma, the WBCs comes to the margin. Rolling occurs with the help of certain molecules which are called as selectants. Adhesion, which helps, adhesion occurs, is otherwise called as peak firm adhesion. This occurs with the help of certain molecules called as integrins. Then you have transmigration, that is, they break the basement membrane and comes out. This occurs with the help of certain molecule which is called as PKM1 or CD31. Then finally you have phagocytosis. This phagocytosis occurs with under 3 steps, that is recognition and attachment, engulfment, killing or degradation of the ingested material. So recognition and attachment. This is initiated by recognition of the particles by receptors expressed on the surface of WBCs. See, for example, we can take this diagram. So the microbes bind to phagocyte receptor. There are certain receptors on the phagocytes which bind certain receptors which are present on the surface of WBCs. So there are certain elements which makes these WBCs a tastier target for phagocytosis. What are these elements? These elements are called as obsonance. I'll come see. The initiated by recognition of particles by receptors expressed on the leukocyte surface. The efficiency of phagocytosis is greatly enhanced when opsonized by specific proteins. These proteins are called as obsonins. Obsonins are the proteins which bind to the surface of the bacteria, which makes it a tasty target for the WBCs, which helps in increasing the efficiency of WBCs for phagocytosis. So now let us see what are all the major obsonins we know of. First, Fc fragment of immunoglobulin antibodies. What is Fc fragment of immunoglobulin? You all know immunoglobulin. So the immunoglobulin is nothing but antibodies. heavy chain, light chain, FC fragment and all. So the FC fragment of immunoglobulin antibodies gets attached to the surface of bacteria. So there is certain receptors for these FC fragments of immunoglobulin on the leukocytes. So whenever the leukocytes come in contact with the bacteria, there is FC fragment which is attached to the surface of bacteria. This FC fragment is obsonant. So it immediately identifies this FC fragment. The WBCs immediately identifies these FC fragments and it engulfs this. Okay. And another is C3b, complement breakdown products, C3b, C4b, C5b. These are all the complement breakdown products that also gets attached to the surface of bacteria. And there are certain plasma lectins that also gets attached to the surface of bacteria. So these are called as obsonance and the process is called as obsonization. Obsonance are nothing but the elements which coat the bacteria and they make the bacteria and tasty target for the WBCs. The obsonance or FC fragment of immunoglobulin, C3b, C4b, C5b breakdown products and plasma lectins. Okay. See? So there are certain obsonance which are present on the surface of bacteria which binds to the surface of WBCs. After binding, what happens? There is engulfment. Phagocyte membrane zips up around the microorganisms. So there is engulfment of these microorganisms inside the cytoplasm of the WBCs. So that is called as phagosome. There is a membrane surrounding these WBCs that is called as phagosome. Phagosome is nothing but the bacteria along with the membrane which is called as phagosome. So, there is a lysosome. It is a cellular organelle which contains certain elements which aids in killing the bacteria, killing the microorganism. So, after phagosome is ingested microbe comes inside the cytoplasm, the lysosome comes in contact with the phagosome. They both fuse together to form phagolysosome. They both fuse together to form phagolysosome, which helps in killing of microorganisms by lysosomal enzyme in the phagolysosome. I'll talk about it talk about all these things in detail. So engulfment means binding of a particle to the phagocytic leukocyte receptors initiate the process of active phagocytosis. So there is extension of cytoplasm around the particle to be engulfed. There is complete enclosure of the particle within a phagosome. The limiting membrane of the phagocytic vacuole fuses with the limiting membrane of the lysosomal granules. So there is discharge of granules content into the phagolysosome. So this is a neutrophil, for example, and it is coming in contact with the microorganisms which are coated by the obsonance like IgG obsonance and C3b and lectin. So it recognizes the microorganisms. Then first step is recognition. The second step is engulfment. It engulfs inside a phagocytic vacuole to form phagosome. The phagosome comes in contact with the lysosome. There is degranulation of the lysosomal granules inside the phagolysosome, which aids in killing of the bacteria. The last step is killing and degradation. The last step is killing and degradation. Killing and degradation within the neutrophils and macrophages occurs by two mechanisms. One is oxygen-dependent method, the other one is oxygen-independent method. What is that oxygen-dependent mechanism? Oxygen-dependent mechanism kills the bacteria with the help of generation of reactive oxygen intermediates. How does these oxygen intermediates comes? This is mainly because of an enzyme which is called as NADPH oxidase. NADPH oxidase is an enzyme which has two components which is present on the surface of the lysosome as well as it has some parts in the cytoplasm. Once the phagocytosis occurs after phagosome formation, what happens? The cytosolic component and the lysosomal component fuse together and this enzyme gets activated. So the NADPH oxidase enzyme reduces oxygen to superoxide anion. Reduces oxygen to superoxide anion. So the superoxide anion is again converted into hydrogen peroxide by spontaneous dismutation. The hydrogen peroxide is reduced to hydroxyl radical with the help of an enzyme which is called as catalase. I'll show you the picture so that it will be better for you to understand. So this is the cytoplasmic oxidase and the NADPH oxidase which is present on the membranes. So this gets activated. So the oxygen is converted into superoxide anion, and the superoxide anion with by spontaneous dismutation is converted into hydrogen peroxide, and this hydrogen peroxide is converted into hypochlorite. Okay. Now there are certain granules which are called as myeloperoxidase. Inside the granules, there are certain enzymes which are called as myeloperoxidase. This myeloperoxidase enzyme, with the help of a chloride ion, converts this hypochlorous into hypochlorite. I'll tell you in detail. The azurophilic granules of neutrophils, that is the myeloperoxidase, in the presence of a halide, converts H2O2 to hypochlorite. Hypochlorite, a potent antimicrobial agent, destroys the microbes by halogenation or by oxidation of protein, and this process is called as H2O2 MPO halide system. It is the most efficient bactericidal system in the neutrophils. So the most efficient bactericidal system in the neutrophils is H2O2 MPO halide system. This is an MCQ, just remember. So now we have dealt about oxygen dependent mechanism. Now we are dealing about oxygen independent mechanism. What are the oxygen independent mechanism? There are certain substances in the leukocyte granules which helps in killing of microorganisms, and there is another protein which is called as bactericidal permeability increasing protein that also helps in killing microorganisms. Lysozyme and lactoferrin. It is an iron binding protein which is present in specific granules. And the most important protein is a major basic protein. Major basic protein is a protein which is present in the granules of eosinophils. What is the color of eosinophilic granules? Crimson red colored granules. So major basic protein is an important component of eosinophilic granules. They aim in killing parasites. They are cytotoxic to many parasites. And the other elements are defensins and elastase that contribute to microbial killing. So you have to, when it comes to killing and degradation, there are two mechanisms, oxygen dependent and oxygen independent mechanism, that you have to be thorough. After killing, acid hydrolysis degrade the microbes within the phagolysosome. So the pH of phagolysosome is between 4 and 5 after phagocytosis. The optimal pH for action of these enzymes is 4 and 5. Now we have completed the phagocytosis. Now we are going to see what are all the defects in leukocyte function. If there are certain defects in any of the process, any of the molecules, that is either in selectin or integrin, you tend to get all these processes, all the cellular events will be affected. So what will happen? This leads to increased susceptibility to infection in all levels of WBC function, that is adhesion, phagolysosome function, microbicidal activity, etc. So these can be genetically inherited or acquired. So the acquired defects in leukocyte function is mainly because of bone marrow suppression, diabetes, malignancy, sepsis and chronic dialysis, anemia, sepsis, diabetes and malnutrition. And genetic, what are the genetic defects? Leukocyte adhesion deficiency 1, this is mainly because defective leukocyte adhesion because of mutation in the beta chain of CD11 or CD18 integrin. Just remember, leukocyte adhesion deficiency 1 happens because of defect of integrin. Leukocyte adhesion deficiency 2 occurs because of mutation in the synthesis of sialylated oligosaccharide, that is the receptor for selectin. Next, there is chronic granulomatous disease. Because of decreased oxidative burst, what happens? The phagocytosis does not happen properly. So the microorganisms or the toxic elements cannot be phagocytosed. So what happens? More and more inflammatory cells like macrophages will come to the site of inflammation or infection in order to get rid of that microorganisms. Gradually, there will be formation of granuloma. That is called as chronic granulomatous disease. Okay, this occurs mainly because of decreased oxidative burst. And some X-linked disease occurs because of phagocyte oxidase membrane component defect. And autosomal recessive leukocyte deficiency. This occurs mainly because of phagocyte oxidase, that is cytoplasmic component. I told there are two components. One is in the cytoplasm, other one is in the membrane. So cytoplasmic component defect causes autosomal recessive disease. And you have myeloperoxidase deficiency. Myeloperoxidase is an enzyme which helps in formation of MPO halide system, H2O to MPO halide system. If there is no myeloperoxidase, that also causes defective kidney. And finally, you have celiac syndrome. This occurs because of decreased leukocyte function because of mutation affecting protein which is involved in lysosomal membrane trafficking. So that's all about today's class. Today's class we have dealt about the cellular events of acute inflammation, which can be classified into 5: marginalization, rolling, adhesion, transmigration, and phagocytosis. I hope you don't have any doubts. If you have any doubts, you can ask. Okay, thank you.